Hello friends, welcome to my YouTube channel, The Microbiology Digest. So we are currently discussing various topics of immunology. So today we'll be discussing another topic that is the about the antigens. So we'll be discussing what is an antigen, what are different types of antigens, and other what are the different other aspects related to the antigen and antibody reaction. So to start with, let us understand what is an antigen. So as we all know, the knowledge of antigen and antibody, it is the basis of the science of immunology. So it is very much essential to understand the antigens or the factors which helps any substance to act as an antigen. So we'll be discussing in detail in the subsequent slides. So in general, when we say antigen, we understand that these are the substances that are capable of inducing an immune response. Means all those substances which can initiate an immune response, they are in general called as an antigen. But if we go into the detail about the definition, exact definition of an antigen, it is defined as the substances or various molecules which are capable of inducing an immune response when they are injected or introduced to a host, suitable host, and they can react specifically with the products that are formed as a result of the immune response. Okay, means an antigen are the substances which can induce an immune response, but it should react specifically with the products that are formed as a result of the immune response. So what are the products of the immune response? So immune response, we know that there are two arms of immune response. That is, the humoral arm will lead to the development or formation of antibody molecules. And these antigens, they should react specifically with the antibody, which are formed by the immune system in response to the introduction of that particular antigen. So again, another term that is called as immunogen. So immunogen, when we say this immunogen and antigen, these two terms are very frequently used interchangeably. Many a times it is used as an immunogen, or it is the term antigen is also used to describe those molecules which are capable of inducing an immune response. But it is very much essential to understand there is a difference between immunogen and antigen. So what is an immunogen? These are the substances which can give rise to an immune response, not only mediated by the antibodies, but the immune response should also be mediated by the specifically sensitized T cells. Means an immunogen, it should be able to induce the immune response, and it is required to be mediated both by antibodies as well as by T cells. Means in general word, to be an immunogen, it should be capable of inducing the immune response by activating both the arms of the immune response. Okay. Now the examples of antigens are numerous. If we say microbes, it may be bacteria, virus, or their components of their cells, they may act as an antigen. And non-microbial source antigens, they include mostly the pollens, egg white, red blood cells. These all, they can act as antigen or immunogen. Now, we have already discussed about the immunogen. Again, there is difference between the immunogenicity and antigenicity. So immunogenicity, what is immunogenicity? These two terms are also very much related. Immunogenicity and antigenicity both are related, but there are differences. So, what is immunogenicity? It is the ability to induce humoral or cell mediated immune response. Means it should, we have already discussed that the immunogen, it should induce immune response by activating both the arms, and that capacity of that immunogens or to induce the immune response by activating both the arms of the immune response, that is the immunogenicity. So if antigens they react with the B cells, so it will produce two subsets. One will be the effector B cells and other subset will be the memory B cells. So these effector B cells will in turn form the antibody molecules. So similarly T cells, when they will interact with the antigen, so there will be again two subsets. One will be effector T cells and the memory T cells. So effector T cells, they will be cytotoxic T lymphocytes. Then helper T cells, they are the effector T cells. So means immunogenicity, it is the capacity to induce an immune response by activating both the arms of the immune response. Now, what is antigenicity? So in the definition of the complete definition of an antigen, we have already discussed, it is the ability to combine specifically with the products that are formed as a result of the immune response. That is the antigenicity. Means to be an antigen, to define completely an antigen, it should have both immunogenicity as well as the antigenicity. Immunogenicity is the ability to induce the immune response, and antigenicity is the ability to combine specifically with the products of the immune response. Okay. Now, all the molecules, whatever we have discussed earlier in the example section, they have the property of immunogenicity. Those particles or molecules, they having the property of immunogenicity, they also possess the capacity of or ability of antigenicity. But reverse is always not true. A substance may be antigenic, but it may not have the ability to induce the immune response, or it may not be immunogenic. Okay. Now there are certain factors. All molecules or all the substances are not antigenic. So they has to meet some of the criterias, or they should have some properties. So what are they? To be an antigen or to be a good antigen, a substance or molecule must have some properties. So these properties are, number one is the foreignness. Then the molecular size. They should have a specific molecular size. Then chemical complexity, structural stability, then degradability. So there are some more factors are also there which are also associated, but principally these five factors are very important to determine the immunogenicity or antigenicity of a substance. Now, what is foreignness? If a substance or to be a good antigen, a substance must be foreign to the body. It should be from the outside source, or it should be foreign to the body. So immune response are not mounted to our body components or self antigens. So if this, there is a strict regulation uh, in the immune system that immune response will not be mounted against the self antigens. If this regulation breaks, then there may be some pathological conditions will arise. So to be a good antigen, a substance or molecule must be foreign to the body. And the degree of foreignness, it determines the ability of the antigen to induce the immune response. Okay. Next factor is the molecular size. To be a good antigen, the molecules must have a minimum molecular size. That is, it should be greater than 1000 daltons. So smaller molecules, they are generally proven to be non-immunogenic or non-antigenic. They do not have the capacity to induce an immune response properly. Then third is the chemical complexity. So to be a good antigen, a substance or molecule should be chemically complex. Then only it becomes immunogenic, or it attains, or it has the ability to induce the immune response if it is a complex molecule. Then next is the structural stability. So this is again another important feature of the antigens. To be a good antigen, the substances or molecules, they should be structurally stable. Why? Because the immune cells, they recognize some specific structures present on the antigens. If these structures are not stable, these antigens or antigenic sites will not be recognized by the immune cells, and these substances will not be able to induce the immune response. Next is the degradability. Degradability means to be a good antigen, a molecule or substance, they should be degradable to smaller particles so that it can be accessible to the cells of the immune system. So there are some examples where although these substances are very complex and they are stable, but as they cannot be degraded to smaller molecules, no immune response is mounted against such substances. Okay, so what are the criteria? Principal criteria. Number one is the foreignness, then molecular size, chemical complexity, structural stability, and degradability. So to be a good antigen, these properties or these characters should be present in a substance or molecule. Then only they will be able to induce the immune response effectively. Now, what are the different types of antigens? So antigens, as we have already discussed, they come from various sources. So these antigens can be classified into different ways. First is based on the solubility. So based on this property, solubility, so we can classify antigens into two major types. One is insoluble antigens or cellular antigens. This example mostly the microorganisms. Then second type is the colloidal or soluble. So here examples are soluble macromolecules which can effectively induce an immune response. Next is classification is based on the origin. Based on origin, we can classify antigens into three different types. Natural means these are all different biopolymers and their complexes which occur in the nature. Then synthetic, these are artificially synthesized. Then the artificials, these are natural antigens which are altered. They comes under the artificial group. Then third type of classification is based on the according to their relationship to the host. Means the relationship of the substance or molecule or the antigen to the host. So based on this, it may be of two types. One is the exogenous. The name itself indicates these are the substances or molecules which uh, originates outside the body of the host. Then second type is the endogenous, which originates within the body. So they comes under the endogenous group. Then based on the type of antigen may be either microbial antigen or non-microbial antigen. Microbial antigen may be either bacteria, virus, or their components, and non-microbial antigens may be dust, pollens. So they comes under non-microbial antigens. Next is the epitope, or it is also otherwise called as antigenic determinants. So these are the sites on the antigen molecule which are specifically recognized by the immune cells. Means these are the sites on the antigen molecule which will be specifically identified by the cells of our immune system, and they will effectively mount an immune response. So these sites or epitopes are also called as antigenic determinants. These are actually the immunologically active sites on the antigen molecule, because these sites they will come directly in contact with the immune cells. These are immunologically active sites. These are maybe these sites or epitopes may be 10 to 12 amino acid long. This is a very small part of the antigen molecule. The size may be it may range from 10 to 12 amino acids only. Then a particular antigen, it may have different types of epitopes. Means different sites may be recognized by the immune cells. So basically there are two types of epitopes are recognized. First one is the sequential or linear epitopes. So these are generally formed by the adjacent amino acid residues, and these linear or sequential epitopes, they do not lost by the denaturation. If these antigen molecules are exposed to some extreme conditions, these epitopes they can be present in the molecule. Then second type of epitopes are conformational epitopes. These are actually formed by the amino acid residues from separate portion of the antigen, and these type of conformational epitopes, they are lost during the process of denaturation. Okay. Next, another term, it is the haptens. Haptens are chemically active substances. They are also chemically active, and they have low molecular weight. And because of this low molecular weight, these substances, they are not capable of inducing an immune response. Means, because we have already discussed that to be a good antigen, all the substances or molecules, they must have a definite molecular weight. Now these haptens, they are also chemically active, but because of their low molecular weight, they are not able to induce an immune response by its own. Means alone they cannot induce the immune response. But because these are active substances, they can induce the immune response if they are conjugated with a with some larger substances. So when we combine these low molecular weight substances with some other molecules, usually proteins, it will increase the molecular weight, and they will be able to induce the immune response now. So these substances which cannot do so, which cannot induce the immune response, they are called as haptens, and the conjugated molecule, attached molecule, they are called as carriers. So when hapten is conjugated with a particular protein, this combination is called as hapten carrier conjugate. Hapten carrier conjugate, when we inject to a particular host, there will be three types of antigenic determinants will be generated. First type of antigenic determinant will be against the hapten determinants. Hapten will have some specific antigenic determinants. So some responses will be directed towards the hapten determinants. Then some response will be mounted against the antigenic determinants of the carrier, and some immune response will be mounted against the combined epitopes or antigenic determinants of the hapten as well as the carrier molecule. Okay, so example of hapten, it is dinitrophenol or IV plant poison. These substances, they are chemically active, but they cannot mount an immune response because they are having a very low molecular weight. Okay, then some other type of antigens we are going to discuss. These are very important to understand, but this is not a classification, just a few antigens which are very specific. They have been named because they serve some specific functions. So that is why we are going to discuss one by one. First type is the thymus dependent or thymus independent antigen. So these thymus dependent antigens, they can stimulate B cells for antibody production, and they always need the help of T cells. T cells means which are the they are indirectly dependent on the thymus. So that is why these antigens are termed as thymus dependent antigens. So they are mostly the protein antigens. Protein antigens, they are thymus dependent antigens, and they mostly stimulate the B cells for antibody production. Then second type is the T independent antigens or thymus independent antigen. These antigens, they can directly stimulate the B cells, but in this case they do not require T cell help. So that is why they are called as thymus independent or T independent antigens. So thymus independent antigens are mostly the carbohydrate antigens. Carbohydrate antigens, and examples of these antigens are the LPS, which is present in the Escherichia coli, then flagellin from Salmonella. They are the independent antigens. Now, there are some differences between these two. First one is the nature. Nature already we have discussed. The protein antigens, they are T dependent antigens are mostly the proteins, while the T independent antigens, they are mostly the carbohydrates or polysaccharides. So T cell help is required in case of T dependent antigens, which is not required in T independent antigens. So antibody response in case of T dependent, it is IgG and IgE, but in T independent antigens, it is mostly IgM is produced. Memory cell in case of T dependent antigen, it is present because B cells will differentiate into effector cells and the memory cells. But in case of T independent antigens, they do not produce any memory cells. Epitope type, these are non-repeating epitopes. These are non-repeating epitopes, and in case of T independent antigens, they are repeating type of epitopes are mostly recognized. Okay, so understanding of this difference is very important. Next type of antigens we are going to discuss is the cross-reacting antigens. So from the name itself, we can understand that these antigens, they come from distinct origins, but they have some common or structurally similar antigenic determinants. And because of these similar antigenic determinants, antibodies which are produced against one of these type of antigens will cross-react with the other member of this group. So these type of antigens are called as cross-reacting antigen, and the phenomenon is called as cross-reaction. So there are numerous reactions or examples of this type of cross-reactions are there. For example, we can say about the Well-Felix reaction, which is a typical cross-reaction between antigens of Proteus bacteria and Rickettsia organisms. So this reaction is mostly used for diagnosis of rickettsial diseases. It is an example of cross reaction or cross reactivity. Then third type is the heterophile antigens. So again, it is also a type of cross reacting antigen only, but it is the name given to several groups of antigens which occur in cells and fluids of generally unrelated individuals or microorganisms. So, but these antigens, they are so closely related because they have identical epitopes. Similar epitopes are present, and because of this property, they cross-react extensively with each other. This type of antigens are called as heterophile antigens, and very good example of heterophile antigen is the Frostman antigen. Frostman antigen is a very good example of heterophile antigen. Then number 4 is the mitogens. Mitogens, these are the substances which can activate lymphocytes for proliferation. Means these substances, they have the capacity to activate the lymphocytes, and because of this activation, lymphocytes will undergo proliferation or division. Okay, so there are several examples of mitogens. These mitogens, they are mostly from plant and animal sources, and sometimes they are also termed as lectins. Okay, some of the examples commonly used mitogens: phytohemagglutinin. This in short it is written as PHA. The source of this mitogen is the red kidney bean, and it can activate the T cells. This is a T cell mitogen. Then second is the concavalin A. It is also called in short it is written as CONA. It is from jack bean, and this is also a T cell mitogen. Then third one is the PWM or porkweed mitogen. This is from roots, collected from roots of Phytolacca americana. Then this is also a T cell mitogen. Then another example is the LPS or lipopolysaccharide from Escherichia coli. This is a B cell mitogen. Okay. Another type of antigens frequently we encounter, they are called as super antigens. So these antigens, they are somewhat different because they can simultaneously bind to the variable beta domain of the T cell receptor and the alpha chain of the MHC class 2 antigen presenting cell. Okay, MHC2 class 2 molecule on the antigen presenting cells, and they can very tightly, they can link the T cell receptor and the APC or antigen presenting cell to trigger a T cell division. These type of antigens are called as super antigens. Means these antigens, the binding of these type of antigens, they are somewhat different to the conventional antigens because they can simultaneously bind to the variable beta domain of the T cell receptor and the alpha chain of the MHC class 2 molecule present on the antigen presenting cell, and they can give a vigorous response for T cell activation. So these type of antigens are called super antigens. So super antigens, they can come from microbial source mostly, as well as uh, for example bacteria and viruses. And they super antigens, they require in a very low concentration, and they are effective in inducing a vigorous, powerful T cell response. Then super antigen responses are not MHC restricted. So what is MHC restriction we will discuss in our subsequent discussions. But super antigens, they are responses to the super antigens are not MHC restricted, but presence of MHC is required, necessary. So examples of super antigens are staphylococcal enterotoxins, then streptococcus pyrogenic enterotoxin, exotoxin of mycoplasma. These are some of the examples of super antigens. Okay. Now neoantigens, these are tumor antigens. These antigens are present in tumors. They are also called as neoantigens. Then autoantigens, these are the antigens which are the self components. When they act as antigens, they are called as autoantigens. This is not a physiological, this is a pathological process. Okay, so this is what I wanted to discuss today. So I hope informations will be helpful for you. If you find it is helpful, please subscribe to my channel and give your comments and share among with your friends so that everyone can get benefited from the informations which are included in this discussions. So with this, thank you very much. Thank you all.